Well, first of all, thanks very much to the organizers for inviting me to speak here today. And I'd like to begin with a picture that shows actually the historical beginning of Alzheimer's disease. This is very much what Alois Alzheimer's first observed and described in his landmark paper in 1906, the appearance of extracellular structures that we now call amyloid plaques and intraneuronal neurofibrillary tangles that are the histopathological hallmarks of the disease that now bears Alzheimer's name. Now, in order to understand the role of plaques and tangles in Alzheimer's pathogenesis, it's important to consider how they form. So, of course, they don't form out of the clear blue sky, but rather they represent the final products of macromolecular assembly processes, whereby individual monomers of the A-beta peptides, or tau, which is the building block of the tangles, first form small oligomers, which then somehow mature into filaments, and it's the filaments that are densely packed in the plaques and tangles. Now, until about a dozen years ago or so, it was widely believed that the plaques and tangles are not only signs of a diseased brain, but in addition, somehow are the causes of the memory, learning, and general cognitive deficits that are so closely associated with Alzheimer's disease. But during the intervening years, it's become quite clear that, in fact, it's small soluble oligomers of amyloid and tau that are not only the most toxic agents in Alzheimer's disease, but also are infectious, at least in a prion-like sense. In other words, toxic misfolded forms of both amyloid beta peptides and tau can self-propagate by catalyzing the misfolding of normal counterparts that may be present in the extracellular space or inside of neurons. So one of the themes that's really emerged and crystallized over the last handful of years is the idea that tau is required for most, probably not all, but most of the adverse effects of amyloid beta. So there are all sorts of insults that can lead to an accumulation of A-beta in the brain, including mutations that cause familial early onset Alzheimer's disease, expression of a normally occurring allele of ApoE, the epsilon 4 allele, environmental insults, and so forth. But in order for this buildup of A-beta in the brain to cause the synaptic dysfunction and neuron death that together lead to impaired memory and cognition, pathways that lead from amyloid through tau have been found to be absolutely required. So what I'm going to talk about for the remainder of my time is how this relates to neuron death. And I'm going to tell you a lot about very detailed molecular mechanisms that connect amyloid and tau to neuron death. And I also want to alert you to John Lazo's talk on Friday morning. John is my friend, UVA colleague, and collaborator, and he's going to tell you how his lab and mine are now trying to leverage some of the findings that I'm about to tell you to find potential therapeutics. So coincidentally, one part of the story that I'm going to relate to you concerns the insulin resistance that's characteristic of Alzheimer's brain and that you just heard about from Suzanne. And this has been known for quite some time, and with very few exceptions, Suzanne's work being notable among them, although we understand that insulin resistance is bad for the brain, very little has been done to describe at a mechanistic level, at a molecular mechanistic level, why that's the case. And I hope to illuminate that issue at least somewhat over the next 15 minutes or so. The other thing that I want to bring up by way of introduction is neuronal cell cycle reentry. So fully differentiated, healthy, normal adult neurons in the brain are permanently post-mitotic. They are in G0. They underwent their final mitotic division during embryogenesis. But in Alzheimer's disease, there's an extended period of time, it may be as long as 10, 15, even 20 years, when large numbers of neurons in areas of brain that will be affected by Alzheimer's disease show all sorts of molecular markers of being back into the cell cycle, at least as far as S phase. But ironically, those neurons do not divide, which might help if such brains were losing neurons. Instead, they actually die. And in fact, it's been estimated that as much as 90% of neuron death in Alzheimer's disease might follow this ectopic neuronal cell cycle reentry. And to put that into context, by the end of a typical long case of Alzheimer's disease, the patient will have lost about 30% of his or her neurons in the frontal lobes of the brain. So this ectopic cell cycle reentry represents what is probably the major cell death pathway in Alzheimer's disease. Now, a handful of years ago, Bruce Lamb and his colleagues published a paper showing that you could recapitulate this ectopic neuronal cell cycle reentry in culture by simply taking primary neurons and exposing them to oligomers, but neither monomers nor fibrils made from A-beta peptides. So with that background in mind, a handful of years ago, a former graduate student of mine, Matt Seward, who now works at Eli Lilly, completed a study that that in retrospect represented our first, or provided us with our first generation understanding of the molecular mechanisms behind this ectopic neuronal cell cycle reentry. So this story begins with taking primary cultured mouse cortical neurons and exposing them to either monomers or oligomers of A-beta, not shown here, also fibrils, the results of which are identical to what we saw with monomers, along with BRDU, bromodeoxyuridine, which can be covalently incorporated into DNA following DNA replication, as would occur in S phase. And as you can see, oligomers of A-beta, but not monomers, cause a large number of nuclei to incorporate BRDU and therefore drive neuronal cell cycle reentry. So this experiment simply reproduced what Bruce Lamb and his colleagues had published a few years earlier, and everything else after that was new, beginning most importantly with the experiment shown on the right, which is exactly the same experiment, except instead of using wild-type neurons, we used neurons derived from tau knockout mice. And as you can see, there's no cell cycle reentry induced by the amyloid oligomers in the absence of tau. With the help of collaborator Eric Roberson, what we showed basically the same thing in a transgenic mouse model of Alzheimer's disease, the HAPP-J20, or simply known as J20 mice, which overproduce a human mutant APP, and these mutations are fully penetrant for Alzheimer's disease in humans, and by the time these mice are 6 months old, their brains are already accumulating large loads of amyloid plaques. As you can see here, there's also a lot of neurons that are positive for cyclin D1, which is a G1 marker. However, when one takes the J20 mice and crosses them with tau knockout mice, the resulting mice that are homozygous for APP and homozygous for a lack of tau, we see no cell cycle reentry whatsoever. So just as in vitro, in vivo, the buildup of amyloid in this, which is normally associated with cell cycle reentry, occurs, but in a manner that depends upon tau. Now, as an interesting sidelight, in an earlier study by Eric Roberson that was published in Science, I think in 2007, he and Leonard Mucke showed that these J20 mice that lack tau, or that for that matter were even haploinsufficient for tau, although their brains were full of plaques, nevertheless they had very few learning and memory deficits compared to the parental J20 line that was homozygous for tau. So what's the mechanism behind this? On a hunch, we began looking for protein kinases that might be involved, and we found 3 protein kinases which, if inhibited with pharmacologic small molecules, could block cell cycle reentry. And those are the kinases in question are CAM kinase 2, FIN, and protein kinase A, all of which, by the way, phosphorylate tau at specific sites and have been implicated in Alzheimer's pathogenesis and in regulation of cell cycle. We also confirmed a report from the Lambert paper that inhibiting mTOR with rapamycin prevented cell cycle reentry. And to follow up on that, our first thought was that, well, amyloid oligomers are interacting with the receptors that sense amyloid, but when we block those receptors pharmacologically, it had no effect. I'm going to get back to this in a few minutes because a big part of our newest story concerns mTOR. So that's a very brief summary of the first paper that we published on the neuronal cell cycle reentry. And this is our first generation model, very low resolution, sort of a framework for the process. Amyloid oligomers, by some mysterious processes, activate these kinases, each of which then must phosphorylate tau at the indicated sites. That leads to neuronal cell cycle reentry and eventually to neuron death. So since that time, we've followed up on a much more expansive study that is now in press at Alzheimer's and Dementia, just appeared on the website a couple of days ago for those of you who may be interested in looking at it. And this is our new model, the second generation model for cell cycle reentry in Alzheimer's disease. It's a very complicated story. I do not have time to go through all of the details, but there are a couple of key parts of this that I do want to share some data with you. And the first concerns the interaction between mTOR and tau. And what I want to emphasize here is what you see is a feedback loop whereby a positive feedback loop whereby tau and mTOR affect each other in a way that seems to be essential for cell cycle reentry. So for those of you who aren't familiar with mTOR, it's a serine-threonine protein kinase that is found in two multi-protein complexes, mTOR-C1 and mTOR-C2, which in the first case is coupled to extracellular receptors and transporters that sense nutrients like amino acids and glucose, and in the case of mTOR-C2 to receptors for insulin and a variety of growth factors. And then collectively, the activation of mTOR-C1 and C2 basically then tells the cell what to do. Should I grow larger? Should I divide? Should I start producing lipids? Should I upregulate or downregulate autophagy? And most importantly, in the context of this talk, cellular proliferation. Should I divide? Now, mTOR-C1 and C2 are connected together by both positive and negative feedback loops in such a way that if either one gets activated, it leads to the activation of the other, which in turn leads to deactivation of the first one and then of the second one as well. Unlike many other protein kinases that, whose activated states can be discriminated by antibodies that can recognize site-specific phosphorylation, we don't have that advantage with mTOR, but for mTOR-C1, we use phosphorylation of 4-EBP and S6 kinase as readouts for elevated mTOR-C1 activity. So shown here are the subunit compositions of the two mTOR complexes, and what I want to emphasize is that Raptor is an essential subunit of mTOR-C1 and Richter an essential subunit of mTOR-C2. So if we knock down either mTOR-C1 or C2 with shRNA, as you can see, either before or after amyloid oligomer application, there's no cell cycle reentry, whereas if we use a scrambled shRNA as a control, amyloid oligomers can induce a huge increase in cell cycle reentry, marked in this case by percent of neurons that incorporate BRDU. In vivo, we found with the help of Salvatore Otto, a collaborator at Arizona State, that some very interesting transgenic mouse model that Salvo developed TG2576 mice. This is another line that overexpresses a human mutant APP and gets plaques and Alzheimer's-like symptoms. That he made a variant of the TG2576 mice that are haploinsufficient for mTOR just in brain. And if we look at 2-month-old mouse brain from either the parental TG2576 line, which is homozygous for mTOR, or the haploinsufficient variant, that looking at 2 markers for cell cycle reentry, Ki67, which marks all nuclei that are anywhere in the cell cycle, G1, S, or G2, or again cyclin D1, which marks G1 alone, we find that there's a huge elevation of cell cycle reentry neurons in the homozygous mTOR animals versus the ones that are haploinsufficient. So in vivo, mTOR C1 seems to be, or rather mTOR itself seems to be very important for cell cycle reentry. This is very relevant to Alzheimer's disease, where steady state levels of mTOR kinase activity are abnormally elevated. So how is mTOR working? Well, what's been known from a number of cell biological studies is that there are 2 principal sites of mTOR C1 action, the plasma membrane and lysosomes. And when mTOR C1 is activated at lysosomes, that inhibits autophagy. Not very much has been known about what happens when mTOR C1 is activated at the plasma membrane, but we decided to test what happens if we force virtually all of the mTOR C1 to either the plasma membrane or the lysosomes by overexpressing Raptor tagged to HRAS20, the plasma membrane targeting domain of HRAS25, or alternatively to Reb15, which targets to lysosomes. So what we found is that when we target all of the mTOR C1 to the plasma membrane, the cells respond just like non-transduced cells, in the sense that there's a big elevation of cell cycle reentry upon amyloid stimulation. However, when the mTOR C1 is forced to the lysosomes, there's no cell cycle reentry. So this represents some kind of fundamental dysregulation of mTOR signaling, because most things, everything other than amyloid that I'm aware of that stimulates mTOR C1 does so primarily at lysosomes and to a lesser extent at the plasma membrane. So it turns out that this stimulation of mTOR C1 this site-specific stimulation at the plasma membrane occurs by some mechanism that requires tau. So here we have, we're looking at phosphorylated EBP proteins. This is a readout of mTOR kinase activity in wild-type cells versus tau knockout cells, looking at mTOR that's been forced to the plasma membrane. Once again, we see an elevation of mTOR activity in the wild-type cells after amyloid application, but not in the tau knockouts. So tau must be present in order for amyloid oligomers to have this preferential activation of mTOR at the plasma membrane. We don't know exactly why yet, but that's something we would like to figure out. So why is mTOR important? What does it have to do with tau? Well, it's not thought that mTOR directly phosphorylates tau, but one of its principal substrates, S6 kinase, does get phosphoactivated by mTOR and is known to phosphorylate tau at serine 262. So we looked at serine 262 phosphorylation in these TG2576 mice that were either homozygous or haploinsufficient for mTOR. And as you can see, there's a huge reduction in tau phosphorylation at serine 262 when only one mTOR gene is present. And in a corresponding model, this is the triple transgenic mice that were fed for 16 months ad libitum with rapamycin-laced food. Remember, rapamycin is an mTOR inhibitor. We see, in addition, a similar reduction of phosphorylation at tau at serine 262. So is this phosphorylation actually important for cell cycle reentry? And the answer is yes. In fact, it's essential. And to figure that out, we went back to tau knockout neurons and re-expressed human tau either in the wild-type form or in a non-phosphorylatable S262A mutation. And what we found is that in the wild-type neurons, after administration of amyloid, there's a huge increase, about a 5-fold increase in cell cycle reentry neurons, but no such increase in the neurons that are expressing the mutated tau that cannot be phosphorylated at position 262. So I finally want to tell you a little bit about insulin signaling. We found that amyloid oligomers, insulin, and amino acids, which would represent a nutrient that can stimulate mTORC1, all had equivalent levels of activation of mTORC1 as marked by 4-EBP phosphorylation. But remember, the insulin and the arginine and leucine, the amino acids, are activating mTORC1 primarily at lysosomes, whereas the amyloid oligomers are activating at the plasma membrane. We found also that if we took neurons and treated them with amyloid oligomers in the presence of either insulin or amino acids, the elevation of cell cycle reentry was abolished. So we can prevent cell cycle reentry with insulin, which should make Suzanne happy, and with amino acids as well. So finally, as you heard from Suzanne, there is evidence that that amyloid oligomers actually drive insulin resistance in neurons by stimulating phosphorylation of IRS at a serine in IRS1. And so putting together that very beautiful paper, along with everything else that I told you about from our lab, that indicates that the ability of amyloid oligomers to suppress insulin signaling in brain is what unleashes the additional toxic potential of those same amyloid oligomers to drive neuronal cell cycle reentry and neuron death. Now, interestingly, at least in tissue culture, everything that you see here occurs by a mechanism that absolutely requires amyloid oligomers and tau, completely independently of their incorporation into plaques and tangles, using soluble amyloid and tau over the course of just a handful of hours. So the story that I just told, I'm going to skip this one. The story that I just told you adds to this selection of prior papers, just a sampling of prior papers that have characterized pathogenic connections between amyloid and tau. And so together, this has prompted us to coin the phrase that amyloid is the trigger and tau is the bullet for amyloid, or in Alzheimer's pathogenesis. And of course, I just sit in my office looking at my computer all day long, typing on a keyboard once in a while, or if not very often. But these are the people who actually do the work. The first part of the story was done by Matt Seward, who's now at Eli Lilly in San Juan, Puerto Rico, as a staff scientist. And Andres Norambuena has done all of the more recent work on mTOR. He's a very, very talented postdoc, and he's looking for a real job now. And of course, we've had lots of help from funding agencies. So sorry for speaking a little bit too long, but thank you for your attention.